Clinical trials are the last step in medical research. They determine whether new treatments make it from bench to bedside. Because they put humans at potential risk, they are heavily monitored and regulated. There are some special design issues with this type of research study. We'll explore some of these. Clinical research can be broadly categorized by the timing of data collection and the participants' comparison conditions. Prospective studies collect new data, whereas retrospective studies use data from the past, either through previous medical records or self-report. Cohort studies are within subjects' designs that look at some future outcome compared to baseline conditions. Case control studies compare individuals of clinical interest with controls on some measure and are between subjects. These are often used to compare the history of individuals with a particular disorder compared to those without. This shows three different approaches to clinical research. On the top is a case control, which collects information that has already happened. Those with and without disease are then compared on this past measure in order to find possible causes. In the middle is a prospective cohort study, where one group is exposed to something in the past or not, and future outcome measures are compared. In a retrospective cohort on the bottom, researchers compare two groups that differ on a particular attribute and look later for measures of disease. In many cases, this can be done using past medical records. We'll focus on a particular type of prospective cohort study, randomized clinical trials. This study compared two different antidepressant drugs, the selective serotonin reuptake inhibitors sertraline and citalopram, against placebo on a number of measures, including the Hamilton Depression Rating Scale shown here. The figure on the left shows average change from baseline for each of the three groups of participants over time, downward indicating improvement. Clearly, both SSRIs produced an improvement in symptoms, although the placebo treatment also resulted in a reduction in depressive scores. The figure on the right shows percentage of participants whose Hamilton depression scores declined by 50% after treatment, taken as a measure of remission. Over half the participants in both the citalopram and sertraline groups showed a 50% reduction, and again, almost 40% of the placebo also showed this degree of improvement. Placebo effects are well-known participant expectancy effects and are almost always Placebo effects are the best known participant expectancy effects, and so placebos are almost always used in clinical drug trials. Rigorous studies are usually double-blind, so that neither researcher nor participant knows what treatment was received until the data has been collected. However, sometimes it doesn't seem to matter if the participants know they're taking a placebo, as shown here in this study of irritable bowel syndrome, or IBS. Neither group received any therapeutic treatment, but half were told they were getting Neither group received any therapeutic treatment, but half were told they were getting a placebo pill and that this actually led to an improvement. COVID changed our lives in many ways. I never thought I'd have to match a non-surgical mask with my outfit every day. Luckily, my mother, shown in the middle with an armload of her homemade masks, has tons of quilting fabric, so my whole family got an assortment to wear. Luckily, my mother, shown in the middle with an armload of her homemade masks, has tons of quilting fabric, so my whole family got an assortment to wear. I wore her masks to get my Moderna shots, the first on the left at Lawton's and my second on the right at the Nova Center. The COVID pandemic has resulted in an explosion of interest in clinical trials, with the race to create and improve vaccines. So, like my mom, I decided to fight COVID too, so I enrolled in a COVID vaccine clinical trial. Before I had either of my official vaccine shots, I enrolled in this study of a new vaccine, VB1-2902A. This summary table shows the possible groups I'd be randomly assigned to. I had my first visit in April of 2021 and signed the 18-page consent form. Then in November of the same year, I was told which group I was in when the study conditions were unblinded. It turns out I got 2 shots of the new vaccine. This trial was double-blinded, placebo-controlled. The person who popped in to give me the shots was not the same person who collected my data. This was done in case I improved because I thought I was getting an actual vaccine. A typical visit involved a COVID test, a pregnancy test, and a lot of blood collection. This shows the timeline and outcome measures, including various things in my blood, but I also filled out a daily diary about pain and possible symptoms after each experimental injection. The care and rigor used in designing this randomized clinical trial is clear from the amount of work involved. A well-designed randomized clinical trial is powerful since it allows researchers to test the benefits of new treatments like vaccines and drugs. There are a number of methodological issues associated with clinical research, but we'll consider 2. As with any study using living participants, there's a risk of using a biased sample. Selection bias is when eligible participants are excluded in some non-random way. For instance, people with high socioeconomic backgrounds might be more likely to enroll in studies and have the means and time to commit to research participation. My involvement in the COVID vaccine trial involved 9 one-hour visits and 2 phone calls. The SSRI clinical trial by Stahl required multiple measures over 24 weeks. Randomized clinical trials looking at testing treatments like this usually required continued participation, so those that don't adhere to the treatment regimen might differ in many ways from those who do. Therefore, loss through attrition can lead to selection bias, even with a good original sample. Another issue with clinical research is measurement bias, which is of course a problem with any type of scientific research. There are many sources of measurement bias, especially in studies that rely on retrospective self-report. This study asked people with and without rheumatoid arthritis about their family history. People with rheumatoid arthritis were more likely to report that their parents also had it. This could be interpreted as a possible genetic link. You'd expect people with the disease might pass it on to their kids. However, the participants in this study were siblings, so there should be no differences in reports between the groups. It's possible those with the disease had better recall of these symptoms and are more likely to have discussed it with their parents. Test yourself. Identify possible sources of error in the study by Stahl et al. 2000. What are some solutions? Clinical trials provide useful information that can have a massive impact on public health, so this research must be designed carefully to avoid pitfalls.